Hello everyone and welcome back. In this lecture we are going to be discussing the evolution of multicellularity, and in the process we'll be giving you an overview of cells and cellular structures, as well as the tissues that are formed when groups of cells specialize and start to work together. We're going to start with a quick introduction to the types of cells and their structures. There are two major categories of cells that we discuss in biology: prokaryotic and eukaryotic cells. Prokaryotic cells are mostly found in organisms like bacteria and a special domain of organisms called the archaeans, which we have discussed before. These are unicellular organisms that tend to live in pretty extreme environments. Bacteria are by far more common, and we have colonies of bacteria living on and in us right now. Prokaryotic cells, such as bacteria, are characterized by a few pretty distinctive characteristics. They have a DNA, but it's not encased inside of a nucleus. It floats around here in the open space, the cytoplasm of the cell. Now, the location where the DNA is found is called the nucleoid. It's just the location of the DNA. The nucleoid isn't its own structure, it's just a place inside a prokaryotic cell. Prokaryotic cells also lack most of the complex organelles that do specialized jobs, which are found in eukaryotic cells. And they also lack most of the organelle membranes that separate a eukaryotic cell into different chambers. Now, eukaryotic cells have all of those things that I just said prokaryotic cells don't have. They have DNA, and it's enclosed inside a structure called a nucleus, which you can see right here. Okay, so this is a cutaway diagram, so you can see inside of the nucleus, and we'll revisit this diagram in a couple of slides. Eukaryotic cells also have complex organelles that are divided up by membranes and are in many cases just made of membranes. Organisms that have eukaryotic cells are all within domain eukarya, which we have discussed before. And that domain can be broken down into these kingdoms: plants and animals, protists, and fungi. All of those groups have eukaryotic cells. Now, eukaryotic cells are found in those four different kingdoms, but there are really just two big groups of cells within the eukarya. Fungi have a modified version of an animal cell. You would discuss those if you were taking botany, and if any of you do move on to take botany, you'll have a fun discussion about how fungal cells differ from both plants and animals. But the most common cells within domain eukarya fall into these two categories: plant cells and animal cells. We're going to discuss them both, but we're going to spend more time on animal cells since, of course, it's a zoology class, and those are the majority of the cells we'll be studying. Now, there are some structures that both plant and animal cells share, and then there are some structures that are unique to one or the other. The plasma membrane is the first structure that's found in both types of cells. It's a membrane that surrounds all cells, and it's what we call selectively permeable, meaning it only allows certain molecules in or out. The plasma membrane is also very flexible. You can see it's this pale white surface on this diagram. It's stretchy and malleable, which is why animal cells can take on a lot of very different types of shapes. Okay? You get a lot of different morphologies of animal cells because this barrier membrane is so stretchy. Plant cells, on the other hand, tend to come in just one of a handful of very geometric shapes. Okay? They tend to be very squared off. And the reason is plant cells don't just have this flexible cell membrane. They are also surrounded by a rigid cell wall, which is depicted in green on my little mini diagrams here. It's an additional barrier. It exists outside the plasma membrane, and it's found only in plants, not in animal cells. Okay? It's made up primarily of cellulose. Cellulose is an incredibly tough carbohydrate. It's indigestible to most animals as well, which is why we're talking about it in a zoology class. The fact that all plant cells are surrounded by this incredibly tough cellulose membrane means it's going to be pretty hard for animals to break through it to get at the nutrients inside the cell. Our digestion lecture is going to talk about how animals have evolved to deal with this cell wall problem. Now, both plasma membranes and cell walls have little holes in them called pores, and those pores allow certain materials to pass through. The pores can be opened or closed, depending on what the membrane or the wall is currently allowing to pass into or out of the cell. The next structure that's found in both of these cell types is called the nucleus. The nucleus houses the DNA. It's surrounded by a membrane called the nuclear envelope, okay, depicted here in purple. Again, this is a cutaway diagram, so they have cut out this portion of the cell, or of the nucleus, so you can see inside. The nuclear envelope also has pores. You can see them here, all of these little dots. Those are pores. And those pores allow molecules to pass in and out of the nucleus. But again, the nuclear envelope is selectively permeable, just like the plasma membrane is. So it only allows certain things in or out. Now, the nucleus contains the DNA, but the DNA is in a special form. DNA in the nucleus is contained in the form of chromosomes. Chromosomes are made of a material called chromatin. So chromosomes are the name of the structure. Chromatin is the name of the material that structure is made from. Chromatin is a mixture of our DNA, which is fiber-like or thread-like in its normal state, and also little balls of protein that the DNA is wound around. So if these are our protein balls, they're called histones, if you're curious. DNA gets wound around these balls over and over again until it forms what looks like a rod. That's your chromosome. Okay? Chromosomes make it easier for the cell to move DNA around. The cell's in a nicely packaged little bundle. If the DNA is in a nicely packaged little bundle, it's going to be easier to move around than if it was in its loose, thread-like state. Okay. Now remember, you don't just have one big piece of DNA in your cells. Your DNA is divided up into many smaller pieces, and each one contains unique information. Each of those smaller pieces gets wrapped around these protein histones and turned into a chromosome. So for each piece of DNA, you have a chromosome. Humans have 23 DNA pieces. And this is humans. Which means we'll also have 23 different chromosomes. But remember, since we're diploid, we've got to multiply that by 2, which means we have a grand total of 46 chromosomes, 2 of each type. Now, that's for humans. Keep in mind that different species have different numbers of chromosomes. So not all animals, not even all mammals, are going to have 23 copies or 23 pieces. The next set of organelles we have to discuss make up a system of cooperating structures called the endomembrane system. This system consists of the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and the vacuoles. There's a couple other structures that are also part of this system, but we're not going to talk about them in detail in this lecture. Right, the endoplasmic reticulum is the manufacturing center of the cell. It's where most molecules are assembled, and that includes proteins. If you look at the little red dots here, these are all ribosomes that are embedded on the surface of the endoplasmic reticulum, which is this blue structure. Okay, so that's the ER. Embedded on one part of the ER are ribosomes, which make proteins. There are free-floating ribosomes in the cell as well, but the majority of our ribosomes tend to be located on the rough endoplasmic reticulum. So this is the rough ER. Okay. This set of tubes up here is called the smooth ER, or smooth endoplasmic reticulum. Okay. And it's responsible for manufacturing other molecules, which generally are based on lipids, so fatty molecules. The Golgi apparatus receives molecules that are created by the endoplasmic reticulum, and the Golgi takes those molecules, packages them up, and ships them out to other parts of the cell. This green structure here is the Golgi. And what you're seeing here are little membrane-bound balloons that are being filled with molecules and then sent off to other parts of the cell. These have been cut in half so you can see inside. So you can see they're filled with little molecules that were produced by the ER. Now the Golgi can produce a number of different types of packages to carry around molecules, and sometimes those packages take on their own identities and do specific work. For example, lysosomes. Lysosomes are little membrane-bound bubbles that break down a variety of different materials. And many lysosomes can be produced by the Golgi. They're filled with digestive enzymes, which means when they encounter a waste product, it can be sucked inside of them and then broken down into smaller pieces, which are then easier for the cell to get rid of. Lysosomes can also be used to digest food and then release the nutrients from that food into the cell so that other organelles can use them. Another membrane-bound sac that's very common in both animal and plant cells is called a vacuole. Vacuoles tend to be larger, and they serve a range of purposes that mainly involve either the long-term storage of materials or the digestion of materials. Digestion is especially common in animal cells. A classic example of a vacuole used for digestion are the food vacuoles that we saw in paramecium earlier in the course. The next set of organelles we need to discuss are all participants in the process of energy. The next organelles we need to discuss all participate in the process of energy conversion inside of the cells. There are two organelles that provide energy in different ways for eukaryotic cells, the mitochondria and the chloroplasts. Mitochondria are in both plant and animal cells. That's important to remember. All eukaryotic cells require mitochondria. The reason is all eukaryotic cells are going to get their energy from sugars and other food molecules. And the mitochondria have specialized in extracting energy from these big, complicated molecules and converting it into a chemical energy molecule that is smaller and easier for organelles to utilize. That molecule is called ATP. ATP is what powers all cellular processes. All the activity that happens in your cells is powered by this ATP molecule. Now, where do those sugar and food molecules come from in the first place? They have to be generated somewhere. They originate in plant cells, and they're created through a process called photosynthesis. Chloroplasts are the organelle responsible for photosynthesis, and among eukaryotic cells, they're only found in plant and algae cells. Photosynthesis is the process of capturing and converting solar energy from the sun into sugars, and those sugars can then be used directly by the plant. This is really important because it's how energy is introduced into the food chain. Ultimately, all of the food energy that animals consume can be traced back to photosynthesis. It all originates with plants, and ultimately, it all originates with the sun. Our next group of cellular structures help to control cellular shape and also cell movement. A structure called the cytoskeleton, which literally translates to cell skeleton, is what's going to provide structural support for our cells. It's a web of fibers that extend throughout the interior of the cell, and they tend to be somewhat hard to see, but they look like this in our diagram. So I'll go over them in red so they stand out. They are these faint but very distinctive, long, fibrous lines. Okay? So this doesn't look like our skeleton, consisting of rigid bones that are very durable and not at all flexible. Instead, this skeleton has some rigidity, but it's also flexible and allows for a lot of cellular movement. The cytoskeleton has a number of jobs. One of the major ones is it provides support for and guidance for the movement of organelles inside the cell to try to help keep the organelles from running into each other. The cytoskeleton can also, interestingly, be used for movement. It can be really quickly disassembled and reassembled in a different part of the cell to change the shape of that part of the cell. Amoebas are a classic example of this. If you start out with an amoeba that looks like this, but it wants to reach out for some, say, particle of food. Okay? What it can do to reach out for that food particle is start to disassemble its cytoskeleton, say from this side of its cell, and move it to this side, so that slowly the shape of the amoeba begins to change, allowing it to reach out for that particle of food. So cytoskeleton is being taken away from this back area of the cell and reassembled in this front area. And what that does is it pushes out on the plasma membrane in this front part of the cell until the amoeba can grab that piece of food. So slowly, it disassembles the cytoskeleton from the back portion of the cell and then reassembles it towards the front, extending its plasma membrane until it can capture what it's reaching for. There's a particular structure that can be found within cytoskeletons. It's called a microtubule. It's one of the most common components of the cytoskeleton. It's exactly what it sounds like. It's a microscopic tube. They're straight and hollow tubes, and they're made of proteins, and they're used to make some pretty unique structures. Flagella and cilia are two structures that are made of microtubules that actually extend through the plasma membrane and help to move the cell around. So they're connected to the cytoskeleton on the inside of the cell, but they extend outwards into the outer environment. Flagella are pretty distinctive. They're long and they swing in a whip-like motion to propel the cell forward. Usually you only get a few of them, sometimes only one, like in these choanoflagellates. Sometimes you get 2 or 3, but usually not many more than that. Okay. Choanoflagellates and choanocytes are classic examples of flagellated cells. Now, cilia are different. They're more numerous, you get a lot of them, they frequently cover the surface of the cell, and they tend to be a lot shorter than flagella. They help move the cell using a coordinated back and forth motion. Okay? So kind of like waving first one way and then back the other. Okay? Then back the other way again. Okay? Back and forth. They're kind of like oars on a boat. Their coordinated movement pushes against the fluid that the cell is floating in and helps propel it along. Now, cilia can help move an independent cell through its environment. Cilia can also help move materials across the surface of a stationary cell, which is what we see for cells that are part of multicellular organisms. Many of these cells are part of an organ where their job is to move fluid or other substances across the cell's surface. For example, the cells lining the intestines, if I were to draw you a cross-section of the intestines with the cilia from their cells, what you'd see is that the cilia inside the intestines move back and forth in such a way that they're constantly propelling fluid in one direction through the intestinal tube. Cilia are in large part contributing to that forward motion and maintaining movement of fluid through the body. Now, because this is so important, we're going to talk a little bit more about how energy and the chemicals that carry that energy are cycled at the cellular level between plants and animals. So remember, photosynthesis is the source of energy for plants and animals. It's a process that uses solar energy from the sun to build organic molecules, which is a fancy name for mostly sugars. Now, as a byproduct of this, the plants also happen to make oxygen. It's not the goal of the reaction, it just happens to get made as well, which is why we call it a byproduct. In order to capture this energy from the sun in these sugar molecules, plants also need 2 other chemicals. They need carbon dioxide and they need water. Okay. The carbon from the carbon dioxide is what's going to be used to make up the carbons in the sugar molecule. So that's where our actual atoms are going to come from. Plants are going to consume the sugars that they produce for themselves, and they're going to do that using their mitochondria. Animals will consume the plants in order to get at the organic molecules inside those plant cells. Now, both plants and animals are going to convert the energy in those organic molecules into ATP energy by using a chemical reaction called cellular respiration. This happens in both plants and animals. We start out with our organic molecule, our sugar molecule, and then using oxygen, we are going to perform a series of chemical reactions, which in the end are going to produce a whole lot of ATP-bearing energy molecules. However, carbon dioxide and water are also going to be produced. But again, these are byproducts. They're not the goal of the reaction. They just happen to get made by it. Now, you may have noticed that the molecules that are produced by photosynthesis our sugar molecule here and our oxygen molecules here, are actually what will be used in our cellular respiration reaction. The mitochondria are going to need this sugar and this oxygen in order to create the ATP that's necessary to power cellular work. Now, as a byproduct of cellular respiration, the mitochondria is also going to produce water and CO2, even though those aren't the goals of the reaction. Fortunately, though, the water and CO2 are necessary for chloroplasts to be able to capture solar energy and trap it in our sugar molecules. The products of one reaction are the reactants of the other. This is why we say photosynthesis and cellular respiration cycle energy through the environment. They cycle molecules as well. All right, so let's start applying some of this knowledge to some of the organisms we've discussed in class. Remember that our oldest animal phylum, the Porifera, have a cellular level of organization, meaning they don't have tissues. They only have cooperating sets of cells. Remember, they're structurally very simple, in part because of their lack of tissues, and they're probably our most ancient animal lineage. Also recall that they are sessile filter feeders. Sessile means once they land in a place, they stay there for life. They don't move again once they've anchored themselves to a rock when they're juveniles. They then filter food out of the water by running that water through the pores of their bodies and allowing their choanocytes to pick food particles out of that water stream using their flagella. Keep in mind, too, that most poriferans are marine, because the oceans are where animal life first evolved. Their structure is pretty simple. It looks like a porous sac that surrounds an open cavity called the spongiocele. Okay, so that is this open space right in here. Right, that's the spongiocele. They have two layers of cells that make up their body walls. There's the epidermis on the outside, epi for above or out. That functions in protection. And then there are the choanocytes on the inside, which function for feeding. They're separated by a non-living layer of material called the mesosyle. Water from the environment enters the sponge through pores. A single one of these pores is called an ostium. You can see them here. Many of these pores, when referred to collectively, are called the ostia. Water exits the sponge through a large hole on the anterior surface of the sponge called the osculum. Okay? So water flows in and then upwards. Choanocytes filter out food particles as that water flows past them. The sponge holds its structure together using spicules that are embedded in the mesosyle. You can see them here. These yellow spiny structures are your spicules. Remember, the material of the spicule is determined by the class of sponge. Demospongiae has spicules made of spongin, truly just spongin supports. Hexactinellids have spicules made of silica. Calcareans have spicules made of calcium carbonate. Now that we have a foundation in cellular structures, let's talk about how those cells likely came together in our animal ancestors to form truly multicellular organisms. Keep in mind that multicellular life is fairly recent. It's only 550 million years old, which is only about 10% of the Earth's geologic history. Now, there are two different ways that multicellular life might have evolved. The two hypotheses are called the colonial hypothesis and the syncytial hypothesis. The colonial hypothesis states that unicellular protists stayed together after cell division, which formed colonies. So our unicellular protists down here, instead of undergoing cell division, resulting in two copies of themselves, which would then leave and swim off in opposite directions, the copies that were created when this protist would divide just stayed together. So after many, many, many rounds of cell division, a colony of many individual protists was formed. If this colony stuck together, it's possible that cells in different parts of the colony would start to undergo specialization. They would start to develop skills that would allow them to be very, very good at one particular job. And as a result of specializing on that one job, they would lose their talents for doing other things. For example, maybe this particular colony of cells started to have individuals on its anterior surface that became really good swimmers. They devoted most of their cellular space to the organelles and energy conversion reactions that were necessary to generate a lot of force for their flagella. This would allow them to be very powerful swimmers, but it wouldn't leave a lot of space inside the cell for organelles that did other things, like, say, waste disposal or food digestion. These cells gave up their abilities to do all these other tasks so they could become really, really good at locomotion. So we're going to call them locomotor cells. Now let's pretend the cells down here, on the bottom of the colony, gave up their ability to swim. Notice they don't have their flagella anymore. They gave up their ability to swim so they could focus all of their energy on being really, really good at synthesizing food, probably from solar energy. So these guys might be photosynthesizers. They might just be really good at cellular respiration and are converting tons and tons of sugar into ATP. But by specializing on that one task, they gave up their talents for things like movement. These two sets of cells are now dependent on each other. The food synthesizers can't move around in their environment without the locomotor cells, and the locomotor cells can't feed themselves without the food synthesizers. This creates codependence, which is really at the heart of being a multicellular organism. But it's not just enough to have cells that do different things in your body. To be a multicellular organism, you can't be a group of separate individuals anymore. You need to be a single individual who just happens to be made up of many smaller parts. The final necessary step in the evolution of multicellularity is to become a single cohesive organism that reproduces itself as a whole unit. So each individual cell isn't reproducing and then creating new cells that float off somewhere. Instead, all of these cells are contributing genetic material to a specialized reproductive cell called a gamete. That gamete, when it's released, will swim off, find another cell to help it reproduce, and then they will create an organism that is made up of the same cooperating cells as this original colony. So each individual cell in the colony is no longer replicating itself. Instead, they're all working together to produce one special type of cell that carries the information on how to rebuild all of them into a brand new individual who is also multicellular. Our second possible origin of multicellularity is from syncytial protists, and thus it's called the syncytial hypothesis. Remember that syncytial cells are large and multinucleated. meaning they have many nuclei within a single cell. Now, if these big syncytial protists were to develop cell membranes that are internal, okay, dividing up the inside of this cell into separate compartments, each of which has a nucleus, what you've essentially created is the same situation we saw in the colonial hypothesis. We have a group of cells that are all connected to each other, which creates the opportunity for a specialization to occur. Okay. Now, the syncytial hypothesis could account for symmetry, especially bilateral symmetry, because many of the syncytial protists that could have evolved these internal membranes already had basic bilateral symmetry. Okay. So a lot of these ancestral protists may have already been bilaterally symmetric. Likewise, they might have then given rise to radially symmetric animals, or it could be that you had a radially symmetric syncytial protist, which then gave rise to radially symmetric descendants. So these are the different ways multicellularity might have evolved. But that then leads us to the question of how many times multicellularity evolved. Was it once? Was it twice? Did it evolve separately in plants and animals? The answer to that question is likely. Probably plants evolve multicellularity independently from animals. But then, did all animals evolve multicellularity from the same ancestor? Is it a homologous trait? And the answer to that question is, it's unclear. We don't really know. It's possible that several different protist lineages may have developed multicellularity separately, which would make their animal descendants polyphyletic. The trick is, there are large numbers of very, very derived similarities among the animals that suggest a single monophyletic origin for all Animalia. It's kind of hard to have this many really specialized physical and chemical similarities among a group of organisms and not have all those similarities traced back to a single shared ancestor. This is what argues strongly for the idea that animals share a single common ancestor, and the odds are very good that since all animals are multicellular, that common ancestor was multicellular as well. Now, there are several big advantages of being multicellular, which would explain why it potentially has evolved many times across all of the kingdoms of eukaryotic life. It facilitates an increase in size. It's a lot easier to get nutrients into and waste out of a large body that's made of many small cells. The cells individually can act sort of like a bucket brigade, where food particles that are captured outside the cell can be passed from one cell to the next very efficiently and very rapidly, which allows the movement of nutrients into the cell and would likewise allow the movement of waste products out of the cell efficiently and quickly. Next, division of labor. Different cells are specialized for very particular jobs within the cell. Different jobs within the body. Certain cells can perform functions more efficiently than a single cell that has to simultaneously do everything. So if you have particular cells that are just focusing on one task, they can become very, very good at it because it's their sole focus. But if you have cells that have to do every task, they have to perform movement jobs, they have to clean up waste, they have to digest food, they have to patrol for immune system disease invaders. They can't be particularly good at any of those jobs because they have to do them all. Right? So this actually makes your body overall more efficient if you have cells that just get really good at particular tasks and then rely on different cells in the body to do the other tasks. And then finally, longer lives. A multicellular organism has a longer lifespan than any individual cell that makes up its body. A particular cell in your body might only live a few weeks, a few months, maybe if it's a long-lived cell, a few years, maybe even decades. But none of those individual cells will live as long as you as the organism will. You overall will outlive most of the cells in your body, and they will have to then be replaced. That's the magic of longer life expectancies for multicellular organisms. If a single cell dies, it can be replaced without killing the entire organism. And remember, a longer life is going to be selected for via evolution because you're going to be able to produce more offspring the longer you live. However, there are risks associated with being multicellular as well. The biggest one is cancer. Mutations in DNA lead to rogue cells that do not contribute to the welfare of the whole organism any longer. They act more like they're living independently. They reproduce on their own schedule, and they use up resources to maximize their own reproduction. This results in uncontrollable cell replication, and it creates what we think of as tumors. Right? The cells in tumors, or just cancerous cells in general, are behaving as though they're independent, and so they no longer perform their specialized task, and they no longer contribute to the welfare of the body. They also continue to use up body resources, but they don't give anything back. And this can ultimately lead to serious damage or the death of the multicellular organism. All right, now that we've talked about how multicellularity can evolve, let's talk about how we put together a multicellular organism. There are a lot of challenges faced by metazoans, by our higher animals, and many of them have to do with how specialized their structures are. One of the biggest challenges that animals face is the problem of differentiation. Keep in mind that all cells in your body have the exact same DNA, but all your cells have to do unique tasks. So how do you get cells to do a unique job that's different from other cells when all cells have the same instruction manual, when they all have the same DNA? Well, if you think of DNA as being an instruction manual that contains the instructions for how to build every part of the multicellular body and how to do all the jobs that are necessary to keep that body running, think of each individual cell as being told it's only supposed to read 1 or 2 chapters in that instruction manual. It's not supposed to read the whole book. Think of those chapters as being genes. Those genes contain instructions for how to do certain tasks or build certain parts of the body. Each cell is told to only read certain chapters in that instruction manual, which means the cell is only told to activate certain genes. The rest of the genes in that cell's DNA are inactivated. So the cell is only following the directions of a few genes, and thus it's only doing a few tasks. This is what leads to specialization. Some genes are inactivated in cells while others are activated to get that cell to do a unique job. This is what's called differentiation. Okay? Differentiation is the process of specialization occurring. Now, after differentiation, your process cannot be reversed. Your cell is specialized forever. After it's become specialized, it stays that way. Now, early in development, initially, all cells have the same potential to become any type of specialized cell. So remember our protostome versus deuterostome development, spiral versus radial cleavage? Remember that the cells early in development, the 1, 2, 4, even 8 cell stages of our embryos, have indeterminate cell fate. Means the cell doesn't have a set fate yet. It isn't already destined to become part of a certain organ system. But that sets in pretty quickly if you're a protostome and you undergo spiral cleavage. Determinate cell fate sets in right at about the 8 cell stage. So those cells already have a job laid out for them. But if you are undergoing radial cleavage as a deuterostome, you maintain your indeterminate fate much later into embryonic development. Your cells do not have a specific task until much later in development. But as development progresses, all embryonic cells in both protostomes and deuterostomes lose their potential, meaning they become more and more set in what they are going to grow into in the adult organism. Okay? Their fate becomes more and more defined as development progresses. These early unspecialized cells that haven't undergone differentiation, that don't have a specific fate yet, are referred to as stem cells. Now, you keep stem cells throughout your entire life. They just lose their flexibility the older you get. But stem cells continually reproduce themselves. They can either undergo cell division and create 2 new stem cells, or they can differentiate into 1 or more types of specialized cell. When that happens, the stem cell undergoes division, it creates 1 new stem cell to replace itself, and it also creates a specialized cell to do a certain job in the body. Now, there are 3 types of stem cells, and they show up at different times during the development of an animal. The earliest stem cells are called totipotent, and they're the most flexible. Think of it in terms of them having total potential. They can turn into anything. The fertilized egg is a classic example of a totipotent stem cell. It's literally going to give rise to every structure in your body. So that's a lot of potential. Pluripotent stem cells are what show up later on in development. So think after gastrulation, when you start having cells that are set to become either part of your digestive tract, or if they're in the ectoderm, they're going to become part of your integument. They can still grow into a huge number of different cell types, but they can't grow into everything. So in essence, they can grow into just about any cell type except for a totipotent cell. They've had some of their potential taken away. I've had some students think of this as plural. It's still a lot, but it's not total anymore. Okay. Multipotent, you could think of multi as being multiple if that helps you. Okay. Multipotent stem cells are what you get as an adult. Okay. They are our adult stem cells. They can grow into cells of a closely related cell family. So for example, blood or multipotent blood cells can grow into red blood cells. They could grow into white blood cells. They could grow into platelets. That's still a lot of different options, but all of those options still fall into the same family of cell types. They're all blood cells, even though they do different jobs. Now remember, specialized cells have to be in the right place at the right time during embryonic development to turn into a specific structure. So if you want to be part of the integument, you're going to have to be part of the endoderm, if you want to be part of the digestive tract, you're going to have to be part of the endoderm during the gastrulation phase of development. If you're not, if you're part of the ectoderm, you're not going to grow into a digestive organ. You're going to be growing into an integument structure. So location during development matters for cell fate. All right. The next topic we're going to discuss briefly is how multicellularity leads to the development of tissues. Now, we did an entire lab on tissues earlier in the semester, so we're not going to spend much time on this now. Remember that differentiated cells will organize into groups with other cells that perform the same function, and that's key. All the cells that make up a tissue are doing the same job. Tissues will then come together in groups to form organs. Organs then come together in groups to form connected organ systems. And our lectures from this point forward in the semester will focus on these organ systems. There are four major types of tissue, four big categories. Epithelial tissue, which is responsible for lining the insides of hollow structures in the cell, or for covering the outsides of hollow structures in the cell. Glandular tissue is a subtype of epithelial tissue. Okay. Glands produce chemicals. Think of things like sweat, oil, even mammary glands, which produce milk in mammals. Connective tissues provide support for the body of the animal, holding things like bones and muscles together. Muscles, speaking of, facilitate movement of the body, while the nervous system provides control for all of the body's systems, sending instructions to and from the central nervous system. Make sure you know these four tissue types for the exam. All right, that's it for today. Thanks for watching.